isn't per se the same Griffith that is named
Griffith Park? I don't know. That's a good question. I don't know. I'm guessing not, but I don't know the answer to that. Okay, so the Griffith experiment basically explored this research question: can organisms transfer the heredity material horizontally?
What do you mean by that?
I'll explain that right now. So make sure you get this written down. So by this point, we knew that you could pass the hereditary material vertically to your children. Okay? So vertically is from one generation to the next, right? Vertical is up and down. So by this point, we knew that organisms could transfer this heredity material vertically. They could pass on their programming to their offspring. But the question was, that Griffith was exploring, is can they pass it horizontally? That is, could they pass it to somebody in the same generation? Could they pass some aspect of their programming to another individual? Yeah.
You mean like siblings?
Sure, or just a random stranger.
Well, how would you do that?
We'll see. We'll see. You just wait. Okay? And so this is what Griffith explored. And ultimately what Griffith found, and I'll show you the data, but ultimately what Griffith found is that the answer to this is yes. At least in bacteria, that there is a large amount of this programming material that can transfer from one individual to another. That individuals can basically transfer their genetic programming to other individuals. So what Griffith found, and again I'll show you these data, the answer to this was yes. And we'll see how it works. Okay, then we have what's called the Avery experiment.
Girl or boy?
Boy, I think. Emma.
Why does it have to be horizontal? That's not work.
Well, we already knew that it could transfer vertically.
Oh.
The question was, could it also transfer horizontally?
That doesn't make sense because I thought the only way you could do that is having a kid.
Well, Griffith did his work on bacteria.
Oh, so you mean like not?
It's like one bacterium transferring it, it's
You get a pastry dish.
petri dish. Or a pastry dish too.
Okay, you know
Yeah,
what, guys?
yeah. Absolutely. But even, I mean, could they also do the same thing in our gut? Like, could the bacteria that live in our gut transfer a heredity material? And the answer to that is also yes. Avery. The Avery experiment basically said, was DNA the material transferred? There's two R's. Transferred? That. Was it one R or two R? Okay. Was DNA the material transferred in the Griffith experiment? So the Griffith experiment showed that individual bacteria could transfer their programming to other bacteria. And so the Avery experiment asked this question: was DNA the material transferred in the Griffith experiment? Was it DNA that was being transferred from one bacterium to another?
No.
It was.
What?
Yep.
How? I don't understand.
Okay? The answer to this was yes. And so I won't show you the data here, but I will explain to you the data of the Avery experiment. The last experiment was called the Hershey Chase experiment. Hershey Chase. Hershey and Chase. And one of these two researchers was a woman, in case you were going to ask that. So the Hershey Chase, I don't remember which, I think it was Chase. Okay, so the Hershey and Chase experiment, basically, by this point, we had a decent understanding of viruses and that viruses inject something into their host to change the host. Okay? To turn the host into a virus-making machine. Alright? So in the Hershey Chase experiment, they asked this research question. Do viruses transfer DNA to hosts? Do viruses transfer DNA to hosts? Because by this point, Avery is making very convincing arguments that DNA is the material responsible for heredity. But other people are saying there's no way. We don't know very much about DNA. Okay, it can't be what's responsible for it. It's going to be proteins. But Hershey and Chase, they asked this question, do viruses transfer DNA to the host? What do you think they find?
No.
The answer is yes.
Maybe you should stop consuming.
Maybe just like go with your
Maybe
gut
she wants break.
and then flip that answer.
And so after these three experiments, there's basically no doubt that it's DNA that's responsible for the program. That it's DNA being passed from parent to offspring to basically predict what the offspring are going to have. It's DNA. That's what's explaining Mendel's ideas of the particulate hypothesis after these three experiments. Okay, there's virtually no doubt. And I want to show you the data from two of these three experiments, and then I'll explain to you the data from the Avery experiment. Yeah.
I don't know if this will make sense, but so for the Avery one, is that questioning Griffith? Is that only because he's wondering if it happens horizontally, but if it was vertically, would Avery not question that DNA was being transferred?
Well, I mean, it'd be a little bit more complicated because if you're going to do just vertical transmission, you have to basically follow for multiple generations. But if you're doing horizontal, yeah, I mean, you can do something that's very quick, and I'll show you their experiment. And so you could still do it if it was vertical, but it's horizontal allows you to do things very quickly, and I want to show you this. Okay, may I erase these? These are the three hallmark experiments: the Griffith, the Avery, and the Hershey and Chase. Now, I will tell you that in the last exam that you all did, some of the questions that I skipped and didn't pull the sheets for were dealing with these experiments. Okay. All right. So here are the data from the Griffith experiment. Okay. First piece of data is that if you injected mice with a particular strain of this bacterium, we call them the S cells. If you injected mice with these live S cells, the mice died. It gave the mice a disease, and the mice died as a result of the bacterial infection. Okay? So you had some of these bacterial individuals called S cells that are virulent, that they spread disease, they cause disease, and ultimately the mice die. Okay? These are the S type of this bacteria. Piece of data number 2 is that if you inject these mice with what are called the R cells, same species of bacteria, but a different strain, the mice do not die. There's no disease triggered in the mice. Okay, so we've got two different types of the same species of bacteria. The type that causes disease and the type that does not cause disease. Okay? Make sense? Now, the type that cause disease, their offspring are going to cause disease. The type that don't cause disease, their offspring are not going to cause disease, right? Because we know that. The programming is passed from one generation to the next vertically. Now, the question was, can they transfer information horizontally? And so, here's what we did. Next piece of data is if you killed the S cells, so they would heat shock them, they'd heat them up and kill those bacteria, which is part of the reason why we cook food, right? Or why you heat something up when it's been sitting in your refrigerator for a little while, on top of the fact that most things don't taste all that great cold. But so if you heat killed those bacteria and you inject the mice with them, the mice don't die. Okay? The mice don't die. So those S bacteria, they have to be alive to cause their disease. And here's the fourth piece of data, and here's the one that showed that horizontal gene transfer is possible. If you took these heat-killed S bacteria and you put them in with the R-type bacteria, whatever it was in the programming of the S-type bacteria that made them disease-causing could transfer to the R-type bacteria. That initially, without exposure to those S-type bacteria, they did not cause disease, but now exposed even to dead S-type bacteria, now the R-type bacteria basically are transformed into S-type. See, and you see this here, living R cells can be converted or transformed into S cells. There's something in that programming that is capable of transferring from one individual to another in the same generation, even when those individuals are dead. The Trinity. So it's basically like you're recycling.
But weren't they already injected with this? So
Well, so they injected them with a mixture of the heat-killed S cells
yeah.
and the R cells. These were different mice. These were new mice.
Oh yeah. So okay.
So if they injected them with just R-type
They died.
bacteria, no, they survived. Just R-type.
Just S type.
If they injected them with just S-type, they died. And if they injected them with killed S-type, they lived. But if they injected them with killed S-type mixed with R-type, something transferred from even those dead S-type cells to the R-type cells to transform them into an S-type cell.
Okay.
There was horizontal, that is, there's horizontal transfer of information. Emma, same generation. And this is really quick, right? All you do is mix them together in a syringe and you inject it and you're done. Because you don't have to wait for the next generation. It's immediate. Emma.
I have two questions.
Yeah.
So I'm not confused on how the killed S cells can transfer some things that live on.
So what's happening there is a process that's called transformation. And so what we now know, they didn't know at this point, but what we now know is that bacteria will take on DNA in their foreign environment and incorporate it into their genome. And so these S types, these S cells, when they've been killed, they will leak their they'll leak their DNA, right? It's a dead cell, it's going to start decomposing, they'll leak their DNA, and these R-type cells can take in that programming. Yeah, it's a process called transformation. Now, when bacteria are still alive, they can deliberately transfer genetic material, but that's a different process. Kyle and then Trinity. Kyle, Trinity, then Emma.
So if R cells are dead and the S cells are dead, nothing happens.
Correct. If the R cells and the S cells are dead, nothing would happen.
So it's only the S cells that can be dead and then the R has to be alive.
Yes, because you need some live bacteria to take on that genetic material, to take on that
What if
hereditary.
it would switch? They tried dead R cells and live S cells?
Well, that would be fine because that live S cells are always going to kill the mice because they're already virulent. They're already disease causing. Yeah. Trinity and then Emma.
Probably a stupid question. If there's like nothing around the dead S cells and they still leak their DNA, wow.
If there's nothing around it, I mean, there's nothing to take on that DNA.
So it just spread. It just be like.
Well, eventually it'll break down. I mean, DNA doesn't last forever. But yeah, if there are any bacteria to take that on, they will take on that DNA. Emma.
Okay, that's just silly, but like, what's the difference between good bacteria and bad bacteria?
Usually whether it causes disease.
Like what in it causes disease?
Oh, it depends. It depends on the particular bacteria. For some bacteria, it's just because our immune system overreacts to their presence. For some, it's because they make a toxin. So, like, we have different strains of E. coli. Most of them are non-virulent. They don't cause disease, and they actually live happily in our gut. They're happy to be there to eat what we're eating, and we're happy for them to be there as well because they make it difficult for other bacteria to colonize. But then there are some strains that secrete toxins. Trinity. Oh, you didn't have... Oh, I thought you had your hand up. Okay, so the Avery experiment, they looked at this Griffith experiment and they're like, okay, or whatever that material is to determine the programming, that hereditary material, it can transfer in the same generation. Okay, because now it's transferring from dead S cells to live R cells, transforming the R cells into S cells. Okay, and so what Avery did, they asked, it was actually a group of researchers, but Avery asked, is it DNA that's transferring? And so what they did was this, they eliminated all of the proteins from the S cells. After the S cells, after they killed them, they heat killed the S cells still, they removed all the protein, okay, and did what Griffith did, and the R cells still transformed into S cells. Okay? So that they showed that it's not proteins then that are transferring from the S cells to the R cells, because even when we eliminated all of the protein before we exposed it to the R cells, it still transformed the R cells. Okay? Make sense? Does that make sense? And then they did it again where they destroyed all of the protein from the S cells and all of the RNA from the S cells and then exposed them to R cells, and it still transformed the R cells into S cells. So they showed that basically even when you eliminate all of the protein and all of the RNA, that the R cells still get transformed. And so that they showed that it is the DNA from those S cells that's transferring to the R cells. Yeah, Kate.
How would you kill all of protein or all of RNA?
This is a good question. You could do it chemically, knowing how proteins are designed, or you can do it where you just you separate them based on size. Okay, and I don't know exactly how they did it. My guess would be that they labeled proteins and they labeled DNA because of their different chemistries, and then they just removed anything that had the fluorescent label of protein. Yeah. There are enzymes that you can put in that eat protein, and there are enzymes that eat nucleic acids, and so you could just put in a protein-eating enzyme in there, and it'll eat all the protein. Yeah. That's what meat tenderizer does. Sprinkle a little meat tenderizer in there, and it eats the protein, which is why it tenderizes meat. Michelle and then Emma.
Can you explain again, like, what's the difference between transfer the material,
Vertically?
Yeah, vertically.
Yeah, so vertically, a vertical transfer of hereditary material would be you passing it on to your children.
Oh.
Horizontal transfer of hereditary material is you transferring it to someone else in the same generation, not your children. Like it would be like you've got some gene that you think I would really benefit from, and you give me that gene.
Would they?
Yeah.
Would then she lose that gene? Is it like
Sure. Yeah. And so bacteria will usually, Kyle asked a good question, like, is it just a one-way transfer? No. Bacteria will replicate their DNA as they're transferring it to some other individual. It's beautiful. Okay? Here are the data from the Hershey Chase experiment. So what Hershey and Chase did was that they labeled all of the protein in a virus, okay, a particular color, and they labeled all of the DNA in the virus a different color. And then what they did was they allowed these viruses to do what viruses do, to inject something into the host. And what they showed was, by having these labeled a different color, they were able to see what ended up inside of the host. And what ended up inside of the host was the DNA. That the viruses did not inject any of their protein into the host. All they injected into the host was their DNA. And so again, another experiment to show that when you transfer DNA, it changes the programming of the individual. If it's bacteria taking in DNA from the environment, it transforms it, potentially into S cells. If it's bacteria being injected by a virus, it's the genetic material, it's the DNA that's transforming the programming. Okay? And so these three experiments again showed that without a doubt, it's DNA that's responsible for the programming of an organism, for the code, for the storing the information, and making sure that that information is carried out into building the machines. Okay? Do these make sense? Because it's really important to know these three experiments and to have the potential to be able to explain them in an essay question.
Wait, so
Hint, hint,
explain
hint,
Hershey
hint,
Chase
hint, hint.
one of them.
Yeah, and so Hershey and Chase, basically the Avery experiment had showed that it's DNA that's transferring from bacteria to bacteria, bacterium to bacterium. But the scientific community was still unconvinced because we didn't know much about DNA. And so what Hershey and Chase did was said, okay, well, we know that viruses inject something into their host. And when they do, it changes the host into a virus-making machine. So whatever they're injecting is a program. And then they're like, what is it that they're injecting? Are they injecting protein or are they injecting DNA? And what they found was that the viruses are only injecting DNA. They're not injecting any protein. So they're injecting DNA, and that DNA changes the program of the bacterium to turn it into a virus-making machine.
Not like one on the right. It's an odd picture.
I'm sorry. Okay. Any questions? Thoughts? Concerns? Okay. So here's what we're gonna do. So take a look at this. All right. So by this time, there's almost no doubt that it's DNA that's responsible for the programming. That it's DNA that transfers from parent to offspring, okay, or from parent to gamete, right, and then two gametes fuse, and there's almost no doubt that when one bacterium changes into a different type of bacterium, it's because it took on some new DNA program. Okay. Now the question is, what does DNA actually look like? How is it structured? Okay, and so you have a group of 4 individuals. You have a group of 4 individuals, and their names are Maurice Wilkins. Maurice.
Oh my god! Maurice is the thing from
It's just diaries, the dog.
no, you didn't have that.
Rosalind Franklin.
His little assistant. Oh my god, love. What a queen.
Maurice Wilkins, Rosalind Franklin.
Do we need these names written down?
Of course.
Yes.
Well, what did they do?
James
I'm
Watson
gonna watch Madagascar.
and Okay. Okay. So 4 names: Maurice Wilkins, Rosalind Franklin, James Watson, and Francis Crick. Okay, Maurice Wilkins and Rosalind Franklin were experts in X-ray crystallography. And so that's what this is here. You take a very highly focused, very powerful X-ray, you fire it at the material, and as these light waves hit this material, it diffracts them into patterns based on the shape of the material. Okay, so they did this. Rosalind Franklin and Maurice Wilkins, they fired X-rays at DNA, and here's the pattern that they got. Okay, they got this pattern here.
Like a chromosome.
Well, it does look like that, but so this pattern is an indication of a helix. Okay, when you see this X pattern, it means that what you're firing at and what's deflecting those X-rays is a helix. And based on the shape and the width of this, that they knew that it was a double helix.
How?
Because they knew how to read X-ray crystallographies. Crystallographies? X-ray crystallography. And so looking at this, Watson and Crick deduced that not only was this a helix, but that it was a double helix.
Okay, so Maurice and Rosalind found out that it was a helix. And
Well,
then
yes.
Crick
And then
Watson said,
looked at this and said that it's a double helix, and then they used some other data that was being published about DNA to determine some other things. Okay, so Rosalind Franklin and Maurice Wilkins did the X-ray crystallography work. James Watson and Francis Crick looked at this and they knew that it was a double helix, and then they looked at a couple of other things. One, by this point, we knew of the 4 bases of DNA, which are A, G, T, and C, right? So by this point, now they know it's a double helix. They know that DNA is made up of a phosphate group, a sugar, and a nitrogenous base. Okay? So those nucleotides, I'll show you these in a minute. Yeah,
How do you
Lisa.
think they found DNA? Like, the nucleus is so tiny.
I mean, same thing they found anything else, by seeing the impacts of it. I mean, and DNA is, when cells are replicating, DNA is big enough that you can see it, right? We looked at cells a couple weeks ago when we were talking about mitosis, and you could visually see the DNA. Okay. By this point, Shh. Focus. By this point, they knew that A and G were purines and that they were large. These were large nitrogenous bases. That A and G, they're called purines, and that they were large, bulky nitrogenous bases.
And this is still Maurice and Rose.
No, this was done by other work. Maurice Wilkins and Rosalind Franklin are responsible for this. And then James Watson and Francis Crick are responsible for synthesizing all of these pieces of information. Trinity.
Wait, what do you mean synthesizing all the pieces of information?
Taking it all and putting it all together to figure out what the overall structure of DNA is. T and C are called pyrimidines. And they are small in comparison to purines. Purines are really big nitrogenous bases. Pyrimidines are small nitrogenous bases. Okay? And so, again, James Watson and Francis Crick, they get the bulk of the credit for the discovery of the structure of DNA, although James Watson, Francis Crick, and Maurice Wilkins, all three win the Nobel Prize for the discovery of the structure of DNA. Rosalind Franklin is left out because she died before they were awarded the Nobel Prize, and you can't award the Nobel Prize posthumously. You also can't award it, at that time at least, and maybe now, you couldn't award it to more than 3 individuals. Okay? And so there's, what's that? For the same thing. And so there's some debate as to whether they delayed the awarding of the Nobel Prize until she died.
That's so mean.
Like, okay,
Okay.
we got this on deck, but like, just wait for it.
Alright, so some other bits of information, okay? So here's some information, the overall shape of DNA, it's a double helix. Looking from this, you can tell that. A and G are large, T and C is small. Some other bits of information that James Watson and Francis Crick didn't determine themselves, somebody else did, and they used to put together the structure of DNA, is the number of purines always equals the number of pyrimidines.
See, 1 to 1.
It's 1 to 1. Okay, the number of purines is always equal to the number of pyrimidines. This right here is called Chargaff's rule.
Number of,
The number of purines in a stretch of DNA is always equal to the number of pyrimidines.
Called what?
Chargaff's rule. Chargaff's rule. Discovered by a man named Chargaff.
I would have said.
Yeah.
There'd be like one base missing. Just like, I don't know if it makes sense. Like, if it's like AT and then C with like the G missing, would that continues?
What if?
Yeah, I mean, could that happen?
Sure, but there are error checking mechanisms in the replication of DNA that we'll talk about in just a minute. Okay, so Chargaff's rule, the number of purines is always equal to the number of pyrimidines. And so Watson and Crick use this to basically deduce that a purine is bonded to a pyrimidine in the middle of this double helix. Okay? That a purine is bonded to a pyrimidine. And this makes sense for 2 reasons. One, these are large and these are small. So if you had a stretch where 2 large bases are bonded together, that's going to be wider than having 2 small bases bonded together, right?
can you draw the DNA different?
I'm going to do better. I'm going to show you a representation. I could draw it, but it would not be good. Okay? I'll show you a picture of it. But just think, if you're going to put 2 large bases together, that's going to be bigger than 2 small bases together, right?
Right.
So the only way it makes sense to have a consistent width in this molecule, which this is implying, is if you bind a large with a small. Every position has a large with a small. And that way the width is consistent on the entire length of the chromosome. Okay? The entire length of the molecule. Does it make sense? So it makes sense for that reason, why a purine binds with a pyrimidine. And then for this reason, it also makes sense. If the number of purines are equal to the number of pyrimidines, it also indicates that those 2 are bound together. And that's why there's the same amount of both. Now further, one moment, Emma, is the number of A always equals the number of T's, and the number of G's always equals the number of C's.
Is that also Chargaff?
That's, another part of Chargaff's rule. So not only does a purine always bind with a pyrimidine in the middle, but specifically this purine binds with this pyrimidine, and this purine binds with this pyrimidine. A binds with T, G binds with C. Emma.
Why can't G bind with T and A with C?
Because it just doesn't work. They don't make hydrogen bonds. Yeah. When you go to align them up, it doesn't work. But they didn't know that until they knew that it had to be A with T because the number of A's equaled the number of T's. If you had 4,000 A's, you'd have 4,000 T's. But even so, now afterwards, we know a little bit more about their chemistry, and we know that it wouldn't work. And so here is a molecule of DNA. Here's a double helix. Okay, so we've got both sides, both strands are spinning, are a helix, are a coil. And there's a consistent width everywhere on the molecule because you always have a pyrimidine, which is a small base, bound to a purine, which is a big base. Notice A is bigger than T here. Do you see that? That this bar is bigger than the T, this bar is bigger than the T. For a reason, because A is bigger than T. It's physically bigger. G is bigger than C. It's physically bigger. And so putting a big base with a little base and putting a big base with a little base and doing that at every position ensures that your width is going to be constant along the entire length of your DNA molecule. Emma.
What is like the outside?
The outside is an alternation of, here, let me show you. The outside is an alternation of the sugar and the phosphate of the nucleotide. Okay? So this right here is one individual nucleotide. This right here is the next individual nucleotide. This right here is the previous individual nucleotide. This right here is 2 nucleotides up. Okay, and so every nucleotide is a sugar, a phosphate, and one of these nitrogenous bases. Okay, one of these nitrogenous bases. Now, so what they did, what James Watson and Francis Crick did, was took all of this information, put it all together, and said this is how DNA must be structured. It must be structured so that these backbones, the handrails on this spiral staircase, if you will, are alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And on the inside, the steps of this spiral staircase, if you will, are these nitrogenous bases. And you always have a purine with a pyrimidine, a purine with a pyrimidine, a purine with a pyrimidine. And specifically, you always have an A with a T, and a G with a C, and an A with a T, and a G with a C. It always works that way. Because it's the only thing that you could use to explain this, that the number of A's is always equal to the number of T's. The number of G's is always equal to the number of C's. And now we know it's the only way in which you can actually facilitate these hydrogen bonds to form between the two strands. Yeah, Michelle.
How does the hydrogen works?
How does it work? Yeah, it works basically the same way water molecules are attracted to each other, or water molecules are attracted to other polar molecules. It's because when you have a polar molecule, you have a positive region and a negative region, right? And so the positive region on one molecule is attracted to the negative region on another. That's a hydrogen bond.
Keeps them like together?
Yes, it keeps them together until you do something that can break hydrogen bonds, like heat them up, or just get in there and just disrupt the hydrogen bonds. But yeah, it keeps them together.
Which one is active? Which one is passive?
That's a good question. On this base, you have both a negative and a positive pole. So on this base, you've got a negative region and a positive region. On this base, you have a negative region and a positive region. So it's not like A's are always negative, T's are always positive. It's A's and T's are both positive and negative. They're both polar.
It's like, so there's like 2 pairs of negative and positive, so it goes like.
Yes. So that the negative on this is attracted to the positive on this, and the negative here is attracted to the positive here. And the only way in which you can get these bases in close enough proximity to actually form these hydrogen bonds is to make it to where these strands are anti-parallel. Do you see this term here? So this is telling us that DNA, and this is something Watson and Crick deduced, that DNA is both complementary, that is one strand is lined up perfectly with the strand of another so that they can always bind, the A's with T's, the G's with C's, there's the complementary, but they are also anti-parallel. So do you notice that this strand runs 5' to 3'? And this is naming which carbon is unbound. So when we get to here, this is carbon number 5. So here's carbon 1, 2, 3, 4, and 5. Okay? Do you see those 5 carbons? At this end, it's a carbon 5 that's not attached to another nucleotide. Do you see that? There's no nucleotide attached to that carbon 5. There is to this one. Look at this. Here's 1, 2, 3, 4, and 5. So attached to that carbon 5, there's another nucleotide, right? But at the very end of this, you have a carbon 5 that is not attached to another nucleotide. When we get to the other end, which carbon is it? It's carbon 3. No, this one's already numbered 4. It's convenient. It's carbon 3 that's not attached to another nucleotide. If we go to this one, that carbon 3 is attached to another nucleotide. You see that? This one, that carbon 3 is attached to another nucleotide. But at this end, you have what's called a free carbon 3. And at this end, you have a free carbon what? 5. So here's the 5' end, here's the 3' end. Notice on the other strand, the 5' end's at the bottom, and the 3' end is at the top. They are anti-parallel. The two strands are parallel to each other, but they go in opposite directions. And that's the only way in which you can facilitate making these hydrogen bonds, is for DNA to be complementary so that you can have those bonds between the bases, but it also needs to be anti-parallel. You had a question, Issa?
Oh, that was my question.
Okay.
What was the 5 carbon?
Alright, so does that make sense? Just referring to which carbon does not have another nucleotide, because it's carbons 5 and 3 that are involved in these nucleotide bonds. Okay?
Are we gonna have a break?
Yeah. Maybe? Okay. Any other questions? So we've talked about, now we know DNA is the material responsible for programming. It's the hereditary material. Yes?
Yes.
And we know the structure of DNA. Now we can do some cool stuff. But before we do some cool stuff, we'll take a break. 3 minutes. All of this.
Katie, say
Okay.
great thing.
All right. Katie, what is step 1?
Step 1 is to unzip.
Unzip the 2 strands. What about step 2? This is where it gets a little bit more interesting, Emma.
Copy the information.
Yeah, but there's actually a step you have to do before you can just copy all of the information. So steps 2 and 3 together are copy. But they actually, you have to do that in 2 steps. Do you know the 2 steps, Katie?
Well, do you have to break apart the base pairs?
Well, that's what you do when you unzip it. Yeah, unzipping is breaking apart the 2 complementary bases. Step 2 is to copy a short stretch as RNA. You copy a short stretch, and this is of both strands. Copy a short stretch as RNA, and this is both strands. And the reason for this is there's no machine in bacteria or in eukaryotes that can just start the process of writing DNA nucleotides. That you have to start by writing a short RNA nucleotide. About 10 bases usually. Usually it's about 10 bases. So our average chromosome, the average human chromosome is 150 million bases in length. That's the average human chromosome.
What
Is 100.
chromosome?
The average chromosome is 150 million bases.
There's 10.
Well, we've got.
Let me stop.
Okay, and so this is a lot. This is a lot of nucleotides. So writing 10, not that big of a deal. Emma.
Are all DNA for humans the same length?
No, not all of our genomes are the same length.
Why?
So the original 4 genomes that they published have a great deal of variation because the most common variation among humans is what's called a copy number variant, where you either had a chromosomal deletion or a chromosomal duplication, and so your genome is either longer or shorter than the average genome. Yeah. And so everybody were around 3.3 billion bases long is our genome, but some individuals are like 3.32, some individuals are 3.31, some individuals are like 3.29. Okay, there's some variation there. Not everybody's exactly the same length. Okay?
Sorry.
Hello?
Is Dr. Phillips here?
He is. He's right over here.
Over there.
So write DNA nucleotides to copy both strands. Okay, so that's step 3. Write DNA nucleotides now. After we've written this short stretch of RNA on both strands, now we're going to write DNA nucleotides to copy both strands. Okay? Yeah, to copy both strands. So a DNA molecule is 2 strands, right? As it's being unzipped, both strands are being copied. We call this replication semi-conservative. So after you've replicated it, you take 1 molecule, and now how many do you have? You've replicated it. You take 1, you have 2. Each of those 2 copies has 1 old strand and 1 new strand. Tori.
So I don't know if this has anything to do with it, but like, so if someone's like 7 feet tall,
Yes.
and like, would their DNA still only be 6 feet tall, or would it be
6 feet in length?
Yeah.
Yes. About that.
So everyone's DNA is the same.
It's about the same length. There's a little bit of variation, and this is what Emma was asking. There's a little bit of variation in the length of the human genome, but it's all pretty close. So replace RNA with DNA and fix the nicks.
The next can't.
CK, fix the nicks. Not NIX, but NICKS.
Oh no, Nick's just NIC. That's not.
So we've got a 4-part replication process. We unzip the 2 strands, we copy a short stretch of that new strand as RNA, and then DNA nucleotides copy the rest, and then step 4 is we replace this short stretch of RNA with DNA and fix all the nicks.
How do they just replace it?
That's
Is
a
it like
wonderful
extra
question.
DNA laying around? Like, how does that work?
Yeah, there are tons and tons and tons of DNA nucleotides.
And they just.
Cut out the RNA nucleotides and replace them with DNA. Yep, you got it. Okay. And so I want to show you this. Yeah, Michelle. Shh, I can't hear Michelle, gentlemen.
What's the function of like copying the RNA?
Why do you write a short RNA first and then DNA? Because the machine that does this, the machine that does step 3 is a different machine than does step 2, and this machine cannot attach the first nucleotide. It can only attach where there's already a nucleotide in place. And we have lots of machines that will do this. We'll copy the DNA and write an RNA copy of it. We'll talk about that next week on Monday, that process. But there are a lot of machines that can do this. There are no machines that can attach the first DNA nucleotide. It needs something to already be in place.
So like how does RNA cause DNA to like replicate?
Well, it makes it possible because you couldn't replicate it without it. You have to write that RNA primer. Yes. Now it needs to eventually get cut out, so we've got to replace all these RNA nucleotides with DNA nucleotides. But so it eventually gets cut out, but there's no machine that can start this process and write just DNA nucleotides. It can only attach where there's already a nucleotide in place. Okay. All right. Does this make sense? Maybe?
I think so.
Yeah.
Okay. So this is a 4-step replication process, and it's a good one. And in bacteria, happens at the rate of about 500 bases a second. In bacteria, about 500 bases every second get written as the DNA is being copied. In eukaryotes, it's much slower. It's 10 times slower. It's about 50 bases a second. And we have 300 or 3 billion bases, doing 50 a second takes a little bit of time. Takes a little bit of time to copy our genome. But that's okay. Because the fastest point that we are ever developing when cells are growing and dividing is in utero, but we take a really long time in utero, right? 40 weeks. It's a long time. It's a long time. Not long enough, because, man, the baby comes before you know it, and you're not ready. This is crazy. Alright? Questions? Emma?
Okay, it's not like pregnancy. How can some people like be pregnant for 6 months and they don't know? Like they're still not considered growing.
Some people still get their normal menstrual cycles even while they're pregnant. I mean, they might be a little bit lighter because you obviously can't shed the entire wall of the uterus if you've got a baby implanted in there and growing and developing. And then some people don't get their menstrual cycles even before they're pregnant or not regularly enough to notice. And then some people are just really oblivious. I go, wow, I guess I actually haven't had my menstrual cycle in 6 months. Crazy, didn't even know. Alright? So I want to show you a picture.
I caught him.
Okay, check this out. Check it out. Check it out. All right. So we talked about a 4-part replication process, right? What was step 1? Unzip it. And here's the enzyme doing step 1. Look at that. This is step 1. That's the enzyme that's unzipping the 2 strands, and it's called helicase. Oh! That's amazing.
Righteous.
Okay, now what was step 2? Write a short. Did you say primase?
Yes.
Nicely done. But you write. Step 2 was we write a short RNA copy. That's what this is right here. This short RNA copy, and this is step 2. And the machine that does this is not on this diagram, but Trinity, what's it called? Primase.
Are these like enzymes or proteins?
They are. They're both. They are both proteins and enzymes. Gosh, Trinity, that's wonderful. Okay, yeah, I know. Now I'm really. You know what? Today has been very encouraging to me. Not surprising, because I'm not surprised when you all know things, but I'm encouraged when you all know things. And now I'm excited to grade your exams. I haven't graded them yet. Okay. Alright, what was step 3? Yeah, you take over and write DNA nucleotides, and here it is right here. There's DNA polymerase 3, our machine that is doing step 3. Then it took over. Look at this. Look here. Look here. You see this? What's this, Trinity? No. Did you say you said kinase and not primase?
What?
Yes.
Oh man. Okay. So here is that short stretch of RNA that started the process, written by primase. And then step 3, the DNA polymerase 3 came in and wrote DNA nucleotides, and it's just going to keep trucking along, writing its DNA nucleotides. And what was step 4?
Replace RNA with DNA and fix the mix.
Which is illustrated right here.
Wait, so it's
Oh, it's like trailing?
Is step 3 called polymerase?
DNA polymerase is the enzyme or the machine that does step 3.
So the ligase just kind of like trails.
Step 3? I mean, there's DNA polymerization. I mean, you don't need a name for it. Okay, so here's step 4. Notice step 4, DNA ligase fixes the NICs. And so we've got 2 machines here, DNA polymerase 1 and ligase. DNA polymerase 1 and ligase do step 4. Cut out the RNA cut out that RNA piece and replace it with DNA nucleotides. You notice here there's no more RNA. Like, so here was an RNA primer, here's an RNA primer, here's another one. Look how close they are on this second strand,
That's crazy.
right? And so based on that distance, there should be one about right here, but it's gone. Because step 4's happened, DNA polymerase 1 and ligase have worked together to cut out the RNA nucleotides, replace them with DNA nucleotides, and fix the nicks. Because when you cut stuff out, it leaves the nick, right?
Yeah.
You gotta fix the nicks.
That's interesting.
Okay? So that's our 4-part replication process.
Just a little press.
Now, anybody notice that the 2 strands are different?
Yeah, one's longer.
One of them's longer.
And one
I mean, ultimately they're about the same length.
I just thought it was showing
Different things.
Okay, they're oriented different ways. And why is that? DNA is complementary, but it's also anti-parallel, right?
Hold up, say that again?
DNA is complementary, that is, the base that's in this position here matches the base that's in this position here. Not that they're the same base, but that it's an A and a T, that they can hydrogen bond together. But they're also anti-parallel. Meaning the DNA polymerase cannot be the same, cannot basically be oriented the same way. The way it's oriented on here is going to be the opposite of how it's oriented here, because the 2 strands are opposite. They're anti-parallel. DNA polymerase, you need to write this down. DNA polymerase, whether it's DNA polymerase 1, DNA polymerase 2, which we didn't even talk about, DNA polymerase 3, which is here, DNA polymerase, all of our forms, all forms of DNA polymerase can only write in the 5' to 3' direction. It can only write in that 5' to 3' direction. Also, it can't start the process. That's why step 2 has to happen. What did we say step 2 was again? That we write a short stretch of RNA, and it's written by a machine called primase, and it writes a short RNA piece, because DNA polymerase 1, DNA polymerase 3, DNA polymerase 2, that's not listed here and we'll probably never talk about, they can only write where there's already a nucleotide to attach to. And they can only write in the 5' to 3' direction. Notice here, here's 5' on this end, yes? Here's 3' on this end, that's the direction RNA polymerase can write. But look at this. Here's 5' at this end, 3' at this end, and that's why the DNA polymerase is oriented the other direction. Okay? Because it can only write in this direction. Emma.
So what starts process 3?
Starts process 3?
Yeah.
A primase basically lets go of the strand and allows DNA polymerase to take over. Now I will tell you, okay, go ahead.
Also, so what does RNA do that DNA can't? Or why do you need this stretch of RNA?
Because the machine that writes RNA nucleotides can start the process. There's no machine that writes DNA nucleotides that can put the first nucleotide in place. There's just no machine that can do that. But there are machines that write RNA nucleotides that can write the first base.
So doesn't that mess up the code though?
No, because eventually DNA polymerase 1 will come in, cut out the RNA nucleotides and replace them with DNA.
Okay, is that part of the fixing?
Yeah, that's step 4.
Okay.
Yeah, cut out the RNA and replace with DNA. If you left the RNA in there, it absolutely would change the code. But DNA polymerase 1 comes in, cuts it out, replaces it with DNA, and then ligase fix all the nicks. Okay. So I will tell you this. Our best recent evidence suggests that helicase, both DNA polymerase molecules, and primase are all physically attached together as one big molecular machine. Now this is showing you them not being attached together because that's what makes more sense. right? That, primase isn't even illustrated, but that these aren't physically attached together. But our best evidence suggests that they are all attached together as one big molecular machine, and that they don't even move. What they do instead is pull the DNA through them in the right direction. So you've got this big machine here pulling this strand through in this direction, but pulling this strand through in the opposite direction, pulling it through backwards. And you're like, how does that work? It's a wonderful question, and we're still trying to figure out all of this. But basically, it has to kind of coil this strand of DNA around so that it can pull it through backwards. Okay. So you notice that these strands are anti-parallel. Look how many primers we have here. Over on this strand, we had one primer, right? Let's not use a 1 because that's going to get confusing with this one. Let's just circle this a second time. On this strand, we have a single primer and then a whole bunch of DNA. Okay, so DNA polymerase 1 is only going to need to cut out a short piece here. But look on this strand. We've got 2 in just this short little area. And there should be a third about right there and a fourth about at the end. Do you notice that? So on the other strand, because of this anti-parallel nature of DNA and the fact that this can only write in the 5' to 3' direction, but this entire machine is moving to the right, this one has to be written backwards. And has to be written backwards in short fragments. Do you see that? Because it can't. They're all physically attached together. If they weren't, even if they weren't attached together like this is illustrating, this would have to wait until it gets access to the full molecule. If it was only going to write in one direction. It'd have to wait until the entire thing was unzipped. But what's the average human chromosome length? 150 million bases. It's going to be waiting a long time to get all the way to the end of that.
How big are
One nucleotide is one base. So 1A is one base. 1G is one base. The average human chromosome is 150 million of them. Yeah, Katie.
I don't understand the difference in the direction between the 5' to 3' because they're both still going through the machine the same way, so I don't know why that would make it, why it would need more primers.
They can't go through the machine in the same way. Because if this is going through the machine or if this machine is moving to the right, this is showing you that this machine is moving to the left, right? But you have to remember that step 1 is we're going to unzip, but we don't unzip the whole thing. Look at DNA polymerase is already working while we're still unzipping the rest of the chromosome, right? So step 1 doesn't finish before we start step 2. If it did, then we wouldn't have this problem. We just unzip the whole thing, and one of the strands would start all the way at the right end, one of the strands would start all the way at the left end, and then they'd go. The problem is that would take way too long. Okay? And so while you're unzipping, you have to be replicating. And so this has to wait until it gets enough to access to write that RNA primer, can write some more DNA, and then it'll unzip some more and it'll grab it again, write an RNA primer, take over and write DNA, and then it'll unzip some more and it gets access to more, and again it just keeps writing. Yeah, Emma.
Where do the bases come from?
They're floating in the nucleus by the billion.
So they're just attached? I don't understand how they become part of the DNA.
DNA polymerase takes them and attaches them to the strand.
Oh, okay.
Yep. That's what DNA polymerase is doing, is it takes those DNA nucleotides, it grabs them and attaches them in the correct order. Now how does it know what the proper order is?
They can't bond.
Yeah, it reads this strand. It reads this old strand and it copies it with its complementary base. With its complementary base. So, here we go. This is a great diagram to illustrate that.
It would be great.
Oh yeah, let me erase this. It would be great, you're right. And it will be. Okay, so what James Watson and Francis Crick realized right away, as soon as they came up with the structure of DNA, is if these can only bind together in one way, if the two strands can only attach together in one way, and that's when they're fully complementary, that all of the bases line up, then now we have a way in which we can replicate this. Because as these two strands unzip and we have access to one of these free strands now, DNA polymerase can come in, see that there's a T there and know that the only thing that will bind with a T is what? Is an A. Okay, and then it'll read the next position and it sees a G there. And what is the only thing that can bind with a G? Is a C. So it knows which nucleotide to add. And then it reads this A, and what's the only thing that can bind with A? Is a T. And so that would be the only way that the two strands could have been attached together initially, is for them to be complementary, and it's the only way in which you can copy it. Okay? And it copies it by attaching the only nucleotide that will actually form hydrogen bonds. But it also has to do that anti-parallel to the one it's reading. Right? Because the two strands are anti-parallel. Alright, so we end up with a little bit of a problem. On this strand here. Because replication is moving to the right, and on this strand that's fine, because that's the direction it would normally write, is to the right. But on this strand, the only way it can write is to the left, because it can only write 5' to 3'. And in this way, that 5' to 3' is to the left, and not to the right. Okay? And so this has to wait until you've unzipped enough that it can get enough access that primase can write a short primer, and then DNA polymerase 3 can take over and it can write another little fragment. And then once it unzips further, it can do the same. But we're always going to have, this one is called the lagging strand, and this one's called the leading strand. Because the leading strand is just being synthesized continuously. Okay? DNA polymerase takes over and it'll just keep writing all the way until the end. But in this one, it has to wait until it's opened up enough that it can write some more. Because it can only write moving to the left. Okay? Does that make sense? It can only write moving to the left, but this whole process is moving to the right. So it actually has to write backwards. That's why in this figure they show you DNA polymerase actually moving backwards. In reality, that's not true, but it is the simplest way to think about what's happening. Okay? Any questions, thoughts, concerns? We're going to work with this a lot, and so hopefully it'll just continue to make sense as we deal with it time and 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 time again until it's AP exam time. And then after we take the AP exam, we're really not going to do much. I mean, maybe, I don't know.
We have a party.
Can we have a party?
Yes. Oh, we can
Like
make a
a
biology
party, like a biology.
Biology-themed foods?
No, like we could get like cupcakes
Like
with
a-
like DNA.
Like a figure of a eukaryotic cell made of Rice Krispie treat?
Yeah.
Candy?
We can like get cupcakes and we can do like kids stuff. We can decorate cookies.
So, does anyone see a problem that this is going to create? In addition to having to write this in short fragments, by the way, those fragments have a name. They're named after a person. They're called Okazaki fragments. Okay,
Probably Japanese.
Yeah, I'm guessing. But does anyone else see a problem that's going to happen with this?
Wait, if it's going
So,
5' to 3', they're gonna run into each other. No.
no, because I mean, this is just going to keep going. They've got two
then
strands to deal with.
that's
But
not-
what's going to happen when you get to the end of this linear chromosome?
Then what?
When you get to the very end, this strand's no problem, right? Because this strand will write all the way until the end. But on this strand, the end is going to be an RNA primer. And then when you cut that RNA primer out, you can't replace it with DNA nucleotides because there's no machine that could put the first DNA nucleotide in position.
So do you.
So when you get to the end of this one, what you're going to do is it's going to shorten your chromosome. So when you replicate all of these and you get all the way to the end, this one's going to be the proper length. This one's going to be shortened. It's going to be shortened by the length of the primer, which is about 10 nucleotides.
Wait, and the primer is the RNA?
Yeah, the primer is the RNA. So when you get to the end of this strand and you go to cut out that RNA primer, you can cut it out, but there's no machine that can replace it with DNA nucleotides. Because in order to do that, it would have to attach the first DNA nucleotide, where there's not already something to the left of it to attach it to. So every time you replicate a eukaryotic chromosome, it shortens by about 10 bases. And you're like, oh, that's not that big of a deal. It's a huge deal. And it makes it to where the chromosome gets shorter every time you replicate, and eventually it gets short enough that you start to lose information. And then the cells can no longer function properly.
Oh, is that how we die?
That's one of the primary causes of what we call cellular senescence, when the cells can no longer function properly. And they can't replicate anymore to replace old, worn-out cells. And so you get, listen to this, how many times do you think a cell could replicate before you would end up with a problem?
4. A billion.
So you're going to go 4. Well, remember, we have to start as a single cell, right? Every one of us starts as a single cell
13 trillion.
that then replicates.
42.
It's about 70 times.
Oh.
An individual cell line can replicate somewhere between 50 and 70 times.
I said 50.
But now you have to think about it this way. So every time the cell replicates, you double your number of cells, right?
Oh, exponential growth.
Yes, so 2 to the 50th power is a huge number, right?
Very true.
What is 2 to the 50th power?
That's a fake number.
I mean, that's a thick number. You know, that's actually become my favorite word, is thick.
I like fat with a P.
That. No, see, what I like to do is just, when somebody's like, so 2 to the 50th power is 1.1 times 10 to the 15. That's way more cells than we have in our body. We have somewhere around, I think, 3 trillion human cells, which would be 3 times 10 to the 12th power. Okay, so that's 1,000 times more cells than we have in our body. Okay, but you can replicate somewhere between 50 to 70 times in a cell line before you start losing chromosomal information. Anybody think of this as a problem? You're like, how does that work with generations?
Yeah.
Yeah.
Because that would mean,
Obviously.
anyone think like that would mean that when you make gametes, their chromosomes are going to be shorter than what yours are.
Yeah.
And so every generation is already starting with like a death clock. Right? Like a death clock that's shorter.
But they still
The other.
So we have an enzyme that's called telomerase. Telomerase. And so it recognizes this region. Look at this. Look at it. Look at this. Look at this. Look. Everyone look. Stop writing. Look. So right here, there was an RNA primer. And look how many bases it is. It's 12 bases. They did 12. I don't know why they didn't do 10. But anyways, they did 12 bases. And so that RNA primer got cut out, and you can't replace it with DNA nucleotides because there's no machine that can attach that first DNA nucleotide. Okay? So this chromosome shortened by 12 bases. Uh-oh. Right? And then if you go and you replicate it again, it's going to shorten by 12 more and shorten by 12 more when you replicate it again. But in some cell lines where you need to preserve chromosome length, telomerase comes in, and look at this. It matches the end of this. You see that? It matches the end of that free end. And so now DNA polymerase can come in, write new nucleotides, because there's a nucleotide there to attach to, right? It's writing 5' to 3' this direction on this strand. So it'll come in, write this a little bit longer to give you some room to get some of those DNA nucleotides back.
Wait, so telomerase matches up with the old.
Yes, it's complementary to this, and so it'll match up here. And then DNA polymerase will write a new stretch of DNA, will actually lengthen this strand. So then now we can get a primer in further back, get a few more DNA nucleotides, and then when you cut the primer out, you still lose the end, but it preserves some of the end of that
So
chromosome.
just give me like a buffer.
It doesn't match every single one.
It matches the end of every chromosome, because the end of every chromosome in our genome is exactly the same sequence. Every chromosome in our genome, which there are 23, every one of them ends in the same sequence. So telomerase matches them all. All right, that's it. Have a wonderful day. Enjoy the rest of your Thursday.